Hi guys, this is Dr. Rajeshwar from yrpharmatube. In the previous video, we learnt on the energetics, significance, regulation and fate of glycolysis. If you did not watch the video, hit the I button on top right of this video to watch it. For the other topics of biochemistry and other subjects of pharmaceutical sciences, click on the links given right below this video. In this lesson, we will discuss on the pathway of citric acid cycle. During glycolysis, some of the potential energy in a glucose molecule is extracted to form 2 pyruvates, 2 ATP, 2 NADH and heat. Under aerobic conditions, the pyruvates can be catabolized further by being converted into Acyl CoA, which can enter the citric acid cycle, which is the third stage of catabolism of biomolecules to form carbon dioxide, water and energy. The citric acid cycle has 8 steps and is circular because step 1 is a reaction between Acyl CoA and oxaloacetate, the product of the final reaction in the pathway. Overall, the citric acid cycle results in the formation of 2 molecules of carbon dioxide, 4 molecules of reduced coenzymes, that is 3 NADH plus 1 FADH2 and 1 molecule of GTP. The net reaction is shown here in the figure. The citric acid cycle is also known as the Krebs cycle, which is named after the German chemist Hans Krebs, who worked out the details of the reactions involved in the cycle. The term citric acid cycle is derived from the word citric acid, which is a tricarboxylic acid. The citric acid is often called citrate as the ionized form predominates at biological pH, and the citrate formation is the first and last steps of the cycle, which means the citric acid cycle starts with the formation of citrate and ends with the citrate. In other words, citric acid is consumed and then regenerated by this sequence of reactions to complete the cycle. The citric acid cycle is also called the tricarboxylic acid cycle, in short TCA cycle, in which tricarboxylic acids such as citrate, aconitate and isocitrate participate in the sequence of reactions. The citric acid cycle is the most important metabolic pathway for generating the reduced coenzymes, that is NADH and FADH2, which are necessary for the reduction of oxygen and ATP synthesis in the electron transport chain. The net result of these two closely linked pathways is the oxidation of nutrients to produce usable chemical energy in the form of ATP. Another important function of the citric acid cycle as a source of intermediates for the biosynthesis of amino acids, glucose, heme, etc. Thus, the citric acid cycle is the final common oxidative pathway for carbohydrates, fats and amino acids. About 65 to 70% of ATP is synthesized in Krebs cycle. TCA cycle essentially involves the oxidation of Acyl CoA to carbon dioxide and water. This cycle utilizes about 2/3 of total oxygen consumed by the body. It is the most important central pathway connecting almost all the individual metabolic pathways either directly or indirectly. In eukaryotic cells, the reactions of citric acid cycle occur in the mitochondrial matrix. In prokaryotic cells, such as bacteria which lack mitochondria, the citric acid cycle reactions are performed in the cytosol, with the proton gradient for ATP production being across the cell's surface, that is plasma membrane, rather than the inner membrane of the mitochondrion. The overall yield of energy containing compounds from the citric acid cycle is 3 NADH, 1 FADH2 and 1 GTP. Reaction sequence or the pathway of citric acid cycle. There are a total 8 reactions in this cycle, each of which is catalyzed by an enzyme. All these reactions are divided into 2 parts. The first part of the cycle includes the addition of Acyl CoA to oxaloacetate to form the 6-carbon product citrate, which undergoes 2 separate decarboxylation reactions that give off carbon dioxide. In the second part, functional groups are added and oxidized to regenerate oxaloacetate, which is the substrate that is needed to begin the cycle again. During this process, 4 pairs of electrons are removed from the substrates and passed to carriers, that is NAD plus and FAD, on their way to the respiratory chain. The first step is the formation of citrate, which is catalyzed by the citrate synthase enzyme, a reaction similar to that of an aldol condensation reaction. The citric acid cycle begins with the transfer of a 2-carbon acetyl group from acyl CoA to the 4-carbon acceptor compound, that is oxaloacetate, to form a 6-carbon compound, that is citrate, via Citric CoA. In other words, acyl CoA, which carries the 2-carbon degradation product of carbohydrates, fats and proteins, enters the cycle by combining with the 4-carbon keto dicarboxylate compound, that is oxaloacetate. This results in the transfer of the acetyl group from coenzyme A to oxaloacetate, producing the C6 citrate compound and a free coenzyme A. Thus, there are 2 parts in this reaction. Number 1, the condensation of acyl CoA and oxaloacetate to form Citric CoA, a process catalyzed by the enzyme citrate synthase. The formation of Citric CoA involves the addition of acyl CoA to the carbon-oxygen double bond. And number 2, hydrolysis of the thioester bond in Citric CoA to produce coenzyme A and citrate, which is also catalyzed by the enzyme citrate synthase. In this reaction, a hydrogen atom of the acetyl methyl group adds to the oxygen atom of the double bond, and the remainder of the acyl CoA adds to the carbon atom of the double bond. The reaction is an irreversible reaction. The second step is the isomerization or formation of isocitrate. Citrate is converted to its less symmetrical isomer isocitrate in an isomerization process that involves a dehydration followed by a hydration reaction. Both the reactions are catalyzed by the enzyme aconitase. The net result of these reactions is that the hydroxyl group from citrate is moved to a different carbon atom. Citrate is an achiral compound and isocitrate is a chiral compound with 2 chiral centers, as shown with the asterisk symbol in the structure. Aconitase produces only one of the stereoisomers, that is isocitrate. Citrate is a tertiary alcohol and isocitrate is a secondary alcohol. It is to note that tertiary alcohols are not readily oxidized and secondary alcohols are easier to oxidize. Thus, the next step in the cycle involves oxidation. The third step involves oxidation and decarboxylation reactions. This is the first of 4 oxidation reduction reactions in the citric acid cycle. The reactants are a NAD+ molecule and isocitrate. The reaction, which is catalyzed by the enzyme isocitrate dehydrogenase, is complex. The hydroxyl group of isocitrate is oxidized to a ketone group to form oxalosuccinate by the action of NAD+, releasing 2 hydrogen atoms. 1 hydrogen and 2 electrons are transferred to NAD+ to form NADH. The remaining hydrogen ion is released. Thus, NAD+ is converted to its reduced form, that is NADH. The oxalosuccinate remains bound to the enzyme and undergoes decarboxylation process, thus removes carbon dioxide, which produces the C5-alpha ketoglutarate, which is a keto dicarboxylate compound. These steps yield the first molecule of carbon dioxide and NADH in the cycle. The fourth step is the second redox reaction of the cycle, or the generation of second carbon dioxide molecule by an oxidative decarboxylation reaction, which involves 1 molecule of NAD+, coenzyme A and alpha ketoglutarate. The catalyst is a 3-enzyme system called the alpha ketoglutarate dehydrogenase complex. In this step, a second NAD+ is converted to its reduced form, that is NADH. A second carboxyl group is removed out as carbon dioxide. Coenzyme A reacts with the decarboxylation product succinate to produce succinate CoA, a compound with a high energy thioester compound. This is the second involvement of the coenzyme A molecule in the cycle, the other instance occurring in step 1. Thus, there are three products, NADH, carbon dioxide, and the 4-carbon compound that is Succinyl CoA. Step 5 is the hydrolysis or Acetyl CoA cleavage. In this reaction, Succinyl CoA synthetase catalyzes the hydrolysis of the thioester bond in Succinyl CoA to yield succinate and coenzyme A. The energy from hydrolysis is transferred to the condensation of phosphate and guanosine diphosphate, forming guanosine triphosphate, a high energy compound similar to that of ATP. The reaction is reversible and is an example for substrate level phosphorylation. Magnesium ions and monophosphate are the cofactors that are required in this reaction. In the sixth step, succinate is dehydrogenated by a membrane-bound flavoprotein succinate dehydrogenase to form fumarate. This enzyme also contains iron sulfur centers in addition to FAD. It is the only enzyme of citric acid cycle which is bound to inner mitochondrial membrane. In the conversion of succinate to fumarate, the FAD is reduced to FADH2 and the reaction is reversible. Step 7 is hydration or hydration of a carbon-carbon double bond. A hydration reaction is a chemical reaction in which a substance combines with water. Water is added to an unsaturated substance which is usually an alkene or an alkyne. In this case, addition of water to the double bond of fumarate gives malate, which is a secondary alcohol. The enzyme fumarase catalyzes this reaction. The enzyme is stereospecific, so only the L isomer of the product L malate is produced. The reaction is freely reversible. This reaction is a biological example of the hydration of an alkene to produce an alcohol. And the last step is oxidation or dehydrogenation of malate to the regeneration of oxaloacetate. In this step, malate is converted to oxaloacetate by a NAD+ dependent malate dehydrogenase enzyme. Malate dehydrogenase catalyzes the oxidation of the hydroxyl group in malate to a carbonyl group yielding oxaloacetate. For the third time in the citric acid cycle, an oxidation by a dehydrogenase provides hydrogen ions and electrons for the reduction of NAD+ to NADH and hydrogen ions. The reaction is reversible and one more acetyl CoA can enter citric acid cycle to undergo oxidation. Thus, citric acid cycle operates continuously till all the acetyl CoAs are oxidized. Although the equilibrium of this reaction is far to its left, that is, it favors malate formation, the reaction proceeds to the right since oxaloacetate and NADH, the two reaction products, are removed rapidly and continuously in further reactions. Oxaloacetate so generated allows repetition of the cycle and NADH participates in oxidative phosphorylation. Thus, this reaction completes the cycle. The figure shown here illustrates the overall reactions involved in the citric acid cycle. The table explains the reactions and the enzymes involved in the reactions of the citric acid cycle. This is the list of references followed for the lesson. That's all in this video, the citric acid cycle pathway. In the next video, we will learn the energetics, significance, regulation, and fate of citric acid cycle. Till then, never stop learning and never stop watching my videos. Thank you for watching this video.